Hello, apparently we're live, and we're late as well. That's because I've been having technical difficulties, really bad technical difficulties, but it all seems to be up and running now. I've got with me Cliff Jumper as our tech guru. He's our screen share expert to share what we need. I've got Jade, who's been digging into some of the background, got hog tie, and I have concordance. And we're going to be looking at a specific paper. It's a very interesting paper to dissect. Now, for goodness sake, I have now completely lost the paper.
It's in comments, if you can pull that back up.
I've got it up now. Yeah. It's identification of unique gene expression profile in children with regressive autism spectrum disorder and ileocolitis. Throughout this hangout, we're going to be using the term ASD for autism spectrum disorder and ASD-GI as a patient with autism spectrum disorder and a gastrointestinal problem. Okay. Now, I think what I want to do is set out our terms of reference, our definitions before we actually start. So this is what I'm going to hand over to Hoggles. He doesn't know about this. I'd like Hoggles to describe to us just the basics of ulcerative colitis and Crohn's disease and what's going on there, just the basic symptoms, query underlying causes, etc. So sorry to spring it on you, hog tie.
That's a big ask. That's a big ask. Yeah, I honestly, it's not even that well captured in memory. I remember the symptomology that one of the biggest problems with it is that either of those conditions is that people are prone to smooth muscle spasms. These are remarkably painful situations because they're, as most people know, the colon, both the small intestine and large intestine, are lined with smooth muscle that sort of go in a fashion like this to help the peristalsis, to help pump product along them. And when those muscles, instead of acting in nice coordinated fashion, they sit there and they spasm. It's shockingly painful. It's really easy to treat with drugs. There are lots of drugs that are just straight up spasm blockers, and you take one and you feel, oh, geez, I don't feel there's anything wrong. Of course, the underlying problem is still there. But to be candid, I actually don't have a firm recollection of what's understood to be the causes. I've sort of some vague recollection about ion pumps not working properly with Crohn's disease and that sort of thing. So as Cliff notes on the side, the deer in the headlights look I have on my face is merited. I don't recall any of the. Well, how far do we need to go into the chemistry of it? Because, like I say, it's not something I have to hand.
I think I'm interested in sort of putting forward the difficulty that there can be in identifying these diseases from symptoms alone. And also, they think there is an autoimmune component to them.
Well, with the ulcerative form, I mean, it's actually a lot easier to tell on inspection. But this is not something. And what a funny phrase, on inspection. Inspection means wholly cleaning the bowels with a purgative product. This is not. This is to be distinguished from a laxative that sort of helps your day go a little more smoothly. This is a project that fills the colon with water by drawing it from the rest of your body. Normally there's a certain amount of moisture sort of kept in the bowel to help keep things moving smoothly. But these purgatives actually draw in a shocking amount of water. Your bowels actually inflate with water, and then it actually flushes everything out. So this purgative is quite a remarkable chemical.
It's one of those things where basically the world falls out your bottom.
Yeah. For people, we shouldn't make light of this, but there are many occasions why somebody, if they're going to go for a scope like this, they So I guess it's a little bit funny because there's many reasons why somebody would go for this. If you do, and they give you a product called a purgative, you don't make any plans. You just bathroom you're home for the day. But the reason that has to be done is because you need a clean look at the surface, and the surface there has to be a lavage of some form. And conventionally, they used to actually pump saline in from the bottom and flush things out. And it turns out that's really not necessary. But the point being is it because so like an ulcer is actually a slightly different color in the tissue. The tissue. So I'm talking about the surface tissue, not the muscles in the back, but the actual lining tissue. One thing to keep in mind is you always know your skin is on the outside of your body, right? This is the outside. On the other side of the skin is the inside. With the colon, everything's backwards, right? Everything inside the bowel is technically outside your body, right? You could do this. I'm not suggesting you could. You could swallow a string and have it come out the other side. It stays completely outside your body, even though it's clearly through you. Gruesome, but it's a little factoid. So the inside lining of the colon is actually the outside of your body, and it's subject to getting ulcers the same way that your skin is subject to getting blisters and that sort of thing. And the presence of ulcers on inspection, which is done with a camera that is not terribly comfortably inserted, makes it pretty obvious when you're looking at ulcerations. Yeah. And there's a very distinct pattern. I mean, there's ulcers that are caused through various forms of damage, through dietary problems or aspirin abuse and things like that. These things don't look anything alike at all. If you look at ulcerative colitis, there's striations and lesions that are a lot more obvious that that's the condition you're dealing with. But I mean, in terms of just, I think I'm afraid that my sort of observations on that are sometimes meandering. So I don't want to lose your point, because I think what you're saying is it's really difficult to tell when you're talking to a person, like, how bad is your day going? Oh, I got this terrible gut pain. My bowels are acting in a way that a normal person would not want their bowels to act. It could be any number of things. And in the modern world, with safe colonoscopy being available in most civilized countries, they just say, well, let's take a look at it, and they'll schedule for colonoscopy, because that's the way you can tell. But short of that, and in the years gone by, there was simply no way to tell by interview with a patient. You actually had to get in there and take a look. The blood products aren't that much different, so there's really not much you can do.
There's something that, if I remember correctly, that effectively you've got a pipe, and as food goes through that pipe, it takes the nutrients out and supplies it to the body. And that if someone has an issue with this, they could end up with scar tissue. So if you imagine there's the length of your pipe, and that's how much you would normally get of nutrient absorption. That if you've got scar tissue, let's say, that goes down this far, you've now only got that amount that is able to sustain nutrients.
Mm-hmm. Yeah.
That and water reabsorption is the other thing. There's a very carefully balanced water reabsorption that happens. I mean, you're right. That's a really good point. That the nutrient absorption is important. I mean, the thing is, when you have deficiencies with nutrient absorption, it's generally not a big problem for people in the Western world, because we eat so much food anyway that probably my 8th unpleasant thing I'm going to say during the broadcast so far, but even our poop is nutrient filled. Our bodies simply can't absorb the quantity of nutrients that we put into it. So losing a bit of your ability to absorb some nutrient in the early stages is not really all that severe, but the more urgent problem is the ability to maintain a proper water balance, because too much water, or the failure to remove enough water from the stool leads to diarrhea and or low motility, to where the stool is sitting there idling and like the water's just being pulled out, leads to constipation, which is apparently not a real funny problem for some people. It's actually quite dangerous.
And also dehydration, if the absorption of water from the gut isn't functioning properly.
Yeah.
It's not pleasant, but the symptoms are they're not absolutely specific. And I think that's the point I wanted to get across. Does anybody else want to add anything about the two conditions? Concordance, you want to add anything?
No, but an interesting lead into the clinical background for the controls and the treated groups, whenever you're ready to get into that, because that is provided in the paper, and the way they're treating the disease differs so substantially that I almost think you don't have to go beyond what is sort of supplement table 1 and supplement table 2 in this paper, without seeing that they've introduced so many variables that this is not a paper that can be tracked to do anything really useful. It can't really answer the question being posed.
Yeah.
Because we have a control group, and you pointed this out, where the ages are vastly different.
Yeah, we'll get
but beyond
on to that.
they're not being treated with drugs. The drugs that some of these kids are on, particularly the ASD kids, these are immune modulators and would be expected to produce the effects that they're observing. So I really question the design of this.
Yeah, we'll get into that design.
Yeah, I'll post it.
I think
There's one more.
but ready now. Sorry?
I was going to say, because we touched on the ulcerations and stuff, but we didn't talk about Crohn's disease.
Yeah.
And one of the features of Crohn's disease is enteritis, and for general reference, anything that ends in itis. We always sort of say as a medical joke, like I have wife-itis, my wife annoys me, or whatever. People make fun of that suffix, but in the medical jargon, it has very specific meaning, and it refers to inflammation.
Yes.
So people with Crohn's disease have, they'll say, oh, well, I have enteritis, so referring to the enteric pathway, usually the small intestine, but it can actually include the large intestine as well. And enteritis isn't even a disease. It's simply a redescription of what's observed, and that is inflammation along. There may not, with Crohn's disease, there isn't necessarily ulcers, like it doesn't necessarily have lesions and tears and that sort of thing. I mean, apparently sometimes that can happen with Crohn's, but again, I have no recollection on the sort of the mechanism, like why this happens with them. But it's a very serious problem, and I want to contrast it with an acute disease, not a chronic disease, but an acute disease, and that's a gastroenteritis, where you actually will get a virus. This is sometimes seen on football teams or hockey teams, where they'll share water bottles, and somebody had intestinal flu is the common name for it. So somebody has intestinal flu, they share water bottles with other people on the team, or water bottles they're drinking on the road when they're coming back, and there's some of the viruses on the mouth of the bottle, and all of a sudden everybody's sick and they're throwing up and they've got diarrhea and everything else. In that case, the virus kind of sets up camp in long lines in the tissue, and then the tissue breaks open, and there's very obvious long skinny lesions, and it's incredibly painful. But that inflammation is a result of your body trying to fight off a virus. And doing so at the expense of loss of large swaths of tissue, which have to be replaced. The reason why I mention this is because if you imagine that event, that gastroenteritis, an intestinal flu event, and sort of the acuteness of it, like this is an urgent thing that happens, some people get hospitalized for it, and then contrast it with Crohn's disease, where you get that inflammation, but it's not a battle. Your body, the portions of your body, that particular tissue, acts as if it's in a really, really aggressive battle, and yet there's no opponent in this battle. So there's strong thinking that this is related to an autoimmune disorder, or this is an example of an autoimmune disorder. And I apologize that my recollection is so distant from this. I guess I should have thought I had to read a little bit more about this stuff. But I think in the context of this paper, it gets to be an issue that the immune status of the people, especially with regard to enteric infections and the body's response to suspected enteric infections, turns out to be kind of a big part of the design of this study. So I just wanted to make that point clear too, that aspect of Crohn's disease is sort of chalked up to an apparent battle that isn't really taking place, because it's like any other autoimmune disorder with people sloughing off their own skin and things like that.
Yeah. Anything to add, Jude?
Nope, not at the moment.
Okay, right, so if we go on to the paper now. Now, Cliff Jumper, our technical guru, can you screen share the abstract, the first introductory bit of the paper, the abstract?
Yep, 1 moment.
As you've got it there, and I'll white box you. Right, there we go. That's not it.
This is a private Skype thing. I'm looking for
Oh,
web page links. Where's your bookmark list?
Screens around. Let me try again.
Yeah.
No, that's this.
I know.
Oh, what's going on with it now? No, that's us. What on earth? Was it moved?
Actually, it probably would be us in a different tab. There it was.
Right, is that it?
I've got 4 screens, so sometimes it moves things around.
Right, so there's our abstract, okay? So, the paper. And it's the identification of unique gene expression in children with regressive autism spectrum disorder. Now, it's interesting this because we've had so many people coming out and saying there is some sort of link. And I'm going to read the abstract out for MD who's watching this and they can't see it. Gastrointestinal symptoms are common in children with autism spectrum disorder, and they're often associated with mucosal inflammatory infiltrates of the small and large intestine. Although distinct histologic and immunohistochemical properties of this inflammatory infiltrate have been previously described in this ASD-GI group, molecular characterization of these lesions has not been reported. In this study, we utilised transcriptome profiling of gastrointestinal mucosal biopsy tissue from ASD-GI children and 3 non-ASD-GI control groups: Crohn's disease, ulcerative colitis, and histologically normal, in an effort to determine if there is a gene expression profile unique to the ASD-GI group. Comparison of the differentially expressed transcripts between the groups demonstrated that non-pathogenic, normal tissue segregated almost completely from inflamed tissue in all cases. Gene expression profiles in intestinal biopsy tissue from patients with Crohn's disease, ulcerative colitis, and ASD-GI, while having significant overlap with each other, also show distinctive features for each group. Taken together, these results demonstrate that ASD-GI children have a gastrointestinal mucosal molecular profile that overlaps significantly with known inflammatory bowel disease, IBD, yet has distinctive features that further supports the presence of an ASD-associated IBD variant, or alternatively a prodromal phase of typical inflammatory bowel disease. Although we report qPCR confirmation of representative differentially expressed transcripts determined initially by microarray, these findings may be considered preliminary to the extent that they require further confirmation in a validation cohort. Right, so basically all that gobbledygook says is we've looked at the gene expression profiles in 4 groups to see if these gene expression profiles are all the same or if there's something distinct about each group. So our 4 groups are children with autism spectrum disorder and bowel disease, so that's the ASD-GI group. Children who have not been diagnosed with ASD, but they have been diagnosed with ulcerative colitis. Children who do not have ASD, but have been diagnosed with Crohn's disease. And then the 4th control group, which is children who have not been diagnosed with ASD and who have not been diagnosed with a bowel disease. You've got to bear that control group in mind. Now, given that the abstract actually says, taken together, these results demonstrate that ASD-GI children have a gastrointestinal mucosal profile that overlaps significantly with known inflammatory bowel disease, yet has distinctive features that further supports the presence of an ASD-associated IBD variant. I think we should actually have a look at these groups and see if that statement in the abstract holds up. So if Cliff could now screen share table 1.
Oh, okay, was it so small? Or has it moved over there? Is this the one?
That's the one, yes. Now, this is the first thing that caught my eye about this paper. Right, keep it right there. And we want to look at this very, very carefully. And I think everybody else has noticed this as well, and I've done my video about it. If we look at the groups, the top group, ASD with GI symptoms, and look at the age, mean age, 5.8 years old. The other 3 groups, all of a mean age, around 12. If you look at the youngest in each group, the youngest in the ASD with GI symptoms is 1.8 years. And yet the other 3 groups, the youngest is 4, 5 or 6. And look at the difference between the oldest in each group is 10, in the ASDGI group, but it's 17, 15 and 16 in the other 3 groups. And right away, that's one of those things that it hits you between the eyes like a brick. There is something absolutely distinctive about this group that the paper is interested in, other than the ASDGI, and that is the age range of the group. Now, straight away, I would be looking at that and saying, "This, we need to now just say, no, we can't go any further with this, because we'll never know if the differences are down to the ASDGI or the difference in age."
And let me add to that, that not only are they young, we've got babies here, kids under 2 years old, but these kids are under, they say later in the paper, drugs that are not anticipated to change the mucosa, but some of the things on here are immune modulators, which it's almost, it makes me wonder, it would be like false advertising in the paper to say, none of the kids were on drugs believed to alter the mucosa, but they're on IVIG. They're getting administered intravenous immunoglobulins. They're shooting them with 1,000 donors worth of antibodies in an effort to deplete their immune system, which we know will globally. I mean, my dad, who has MS, is on IVIG specifically to combat his autoimmunity, and one of the consequences of that is gastrointestinal. So, I mean, already we're seeing sort of violations of the way they're describing their population. It really, to me, it's so many variables being interpreted.
Yeah.
It doesn't matter how fancy their statistics or how good their PCA graphs look. I'm looking at it and saying, I wonder if we could tease that out as, whether or not the kids are getting immune modulators. The control population are not only in their teens, but they're also none of them on any known drugs because they're healthy controls. They're all, for some reason, almost all on laxatives, which I guess that explains why they were at a gastroenterologist's office. They probably had constipation problems, and as part of normal checkup, they're getting biopsied for any kind of inflammatory disease, suspected IBD perhaps. What was your thought on that? Why do you think, where did the control population come from? Teens going to a gastroenterologist.
I would like to hit on that control group a little bit later.
Sure.
And for a very good reason. And it's basically the how they're determined to be controls. But I think the concern for me is that straight away, just looking at the age range, this study is going to tell us nothing.
Well, there are ways to tease out, like when there are age differences, but then you have to have a pretty thorough model. Like, I mean, I was puzzled by the age thing at first, but I guess the thing that really threw me off is when you classify. Okay, so we've got something we say there's 25 that are ASD. How do you declare someone in one of the control groups? How do you declare them to be non-ASD?
Yeah,
right.
That's another bit that we can get to.
Well, I'm suggesting there may be a safe point. I mean, if somebody's 30 years old and they have never exhibited symptoms, and this is on inspection, right? You've actually done some testing to say, no, this is, that I can see them including saying, well, we're pretty sure this person doesn't have any sort of autism of any sort, and you wouldn't ever want to make that claim for somebody who's 5 years old. So I kind of thought, well, maybe that's why there's an age separation, but it didn't really seem to be addressed. And in any case, it could have been addressed statistically very differently than just saying, well, we've got some kids in this group and some kids in that group.
The other thing to notice are the sample sizes. We have, in the autism group, we've got 25, 23 of them are male, 2 are female. autism is more common in males, so that could explain it, but it does cause problems when it comes to things like gene expression profiles. Because females express genes that males don't, and males express genes that females don't. Then the other groups, we've got 8, 5, and 15.
None of them are males on the non-ASD with ulcerative colitis.
Yeah, let's get 5 girls to be the ulcerative colitis control group.
Yeah.
That's such an obvious one.
Well, remember now, some of these kids are going to be sort of post-puberty, and the entire ASD group are, I would presume, from the ages, the oldest age is 10 years, probably pre-puberty. So we're looking at hormonal versus, pre-pubescent levels of hormone.
Yeah. And
huge variable.
And we know that gene expression is going to change when they hit puberty. You get genes switching on and off all the time. And the example that I used in my video was the lactase gene, which they think the wild type is actually those who are lactose intolerant. Because lactose tolerance, the only mammal it's seen in, is in humans. And humans are the only ones that actually farm dairy animals. But the lactase gene is switched on until a kid is about 2 years old. And then it's supposed to switch off. And that's when the lactose intolerance starts. They can no longer drink quantities of milk. And that happens throughout all of the mammals. Humans are the exception, and lactose tolerance is the mutated form, shall we say, that's been selected for. So right there, you have an example of a gene in the gut whose expression is age dependent. And how many others are there?
And among the effects of that gene being expressed and having products introduced into the gut is inflammation. And,
I should mention, though, that, again, we're looking at Supplement 1 and Supplement 2 at the very bottom. The special diets are recorded for the ASD, but not for any of the other patients. So for the control,
Becky's prepared something about that.
But
I discovered that as well, and that was, that's very obvious that there's so many things wrong with this paper. Yeah.
Now, I'm going to ask Cliff if what he can do. I'll stop sharing him just now. I'm going to ask him if he can pull up the video I did, and I only want him to find parts of the video. Let's get past table 1 on that. And see if you can find the piece of text at 7 minutes 15.
15. 1 second.
And screen share that just as a still.
One moment. Just finding it. Okay, is that it?
That's it, yeah. Right, let's present to all. Let's see how you do that. Ah, you got it right. And if you look at this, here's something very interesting. Talking about how these patients were selected. That most of the patients in the ASD-GI group were referred for gastrointestinal evaluation with a primary care provider, though some patients were self-referred. And right there, the wee alarm bells start ringing for me. And then the next sentence, all were patients of a single pediatric gastroenterologist, AK, that's his initials. When you look at the authors of the paper, that is Arthur Krigsman. Arthur Krigsman was the pediatric gastroenterologist who was involved with the Andrew Wakefield paper.
Oh, really?
Mm-hmm.
Yeah, and did you mention that he has, I think it's 6 or 7 papers in total in his entire career? Mm-hmm. And maybe for people watching, they don't know what that means, but it basically means that he's a weekend researcher. He most people make it through their PhDs with 1 or 3 or 4 papers by the time they finish their degree, and then they're out 4 or 5. Thunderfoot, for example, has published 5 papers since January of this year. So for a senior PI level person to have 6 publications in total means that's not what they do. They're dilettantes, I guess.
Yeah, and they are medics, and I don't mean that in a derogatory fashion.
Yeah, I was about to step in on that. Not to be unkind about it. There are many health research, what they call academic health science centers, where you have physicians who are there as practitioners / teachers or practitioners / administrators, and then some that are practitioners / researchers. But there's a very big divide in the research focus between somebody who does research and doesn't do the clinical side of it, and the clinician who publishes occasionally. It's certainly not necessarily bad for somebody's career or an indication that they're bad at science if they're a full-time clinician and occasionally publish.
And that was not my intention. I'm not saying that he's a bad person. What I'm saying
let's go back to the Andrew Wakefield thing.
His only publications, actually, I think 3 out of the 6 publications he's managed to produce were co-authored with Wakefield. So that's pretty much been his research experience.
Yeah, and that is another red flag, because I'm seeing a similarity in this paper with one of the problems with Wakefield's paper. There's self-referral. Then there's also the patients of a single pediatric gastroenterologist. Now, going back to, if Cliff could screen share that again.
Yeah, sure.
And I want to be clear, I'm not saying guilt by association here. That's not what I'm saying at all. Right. Right, there we go. Now, it says that they were selected based on a history of normal development for at least 12 months, followed by developmental regression and onset of GI symptoms, okay? And also, for all these individuals, this was their first ileocolonoscopy, and no patients were taking medication thought to alter the histologic appearance of the GI mucosa. But here's the problem. The study isn't looking at the histologic appearance of the GI mucosa. It's looking at gene expression profiles.
Well, they're biopsies, weren't they?
What does that mean?
That you were retrieving cells. Oh, sorry. Okay, I see what you're saying. Maybe I misunderstand what you're saying.
Yeah.
They're actually
you have to remember, I'm not skilled in this kind of thing, so I have no idea what you meant by that.
Okay. Well, there's most of the things you do on a colonoscopy, and regardless how far you go up, they refer to the ileocolonoscopy, where you actually go up to the ileum. So you got that awkward turn at the jejunum, but that's some distance. It's quite, you're downtown, you're uptown when you're doing an ileocolonoscopy. But it's just, you're there for a tour. You're just looking at stuff, right? But there's also extra tools that go with the camera. One of them is a snare to be able to pop off a polyp if there's something that's sticking out. But there's also a little, I'm doing a gesture with my hand. Apparently I'm not shared right now, but it's a little snippy. It looks like a little snippy thing, like the robots, clampy robots from the 1940s movies, sci-fi movies, that you can actually use to tear away a piece of the lining, the tissue. Yeah. And it actually bleeds a little bit. It's a little nick you put in there. But then, where you can, you retrieve that tissue and you can send it off for examination. But the histological examination, we're talking about, you look at it under a microscope. Yeah, I'm sorry, Fiona.
These were actual biopsies. I mean, there's a section here on case selection and biopsy procurement.
Yes, it was biopsies that they took, and what they're saying is that no patients were taking medication thought to alter the histologic appearance, i.e. the microscopic appearance of the mucosa. But
that's what I'm asking. If it's a microscopic experience. Yeah, so this under microscope would look different, but in terms of, I think where I'm confusing myself is I'm thinking about the extra steps in a biopsy when you've got cancerous cells or precancerous cells. For example, you also will where possible, you'll do karyotyping or sequencing or whatever else. So, for some experiments, not for this.
This is just the histology, looking at the actual cell arrangements, etc. But the paper, they're saying that they were not taking any medication thought to alter the histologic appearance. So therefore, the histologic appearance has got to be down to the ASD GI business. But this paper isn't looking at histologic changes, it's looking at gene expression changes. So saying that the medications are not thought to alter the histologic appearance tells us nothing, because they could be altering gene expression profiles. We don't know. And saying it doesn't alter the histologic appearance does not mean that it hasn't affected gene expression profile. So that's not a safety net to allow you to rule out effects due to any medications that they're taking. Because histologic appearance is not gene expression profiles. Now, with gene expression profiles, you're measuring the message that's coming off the gene. You're looking at gene expression, what genes are being expressed. Hold Ty's looking confused.
No, I was just reading the side chat in the comments.
And so, for that reason, I don't think that comment is enough to give them that safety net. And the other thing was 25 consecutive patients meeting these criteria were selected. Now, that little sentence there isn't saying it was 25 consecutive patients. It's saying it's 25 consecutive patients who met the criteria. How many patients didn't meet the criteria? Because you're left thinking to yourself, it could be 3,000. Or it could have been 50. You've got no way of knowing
26.
how representative this sample is of autism spectrum disorder. And so when you're trying to draw the link between the GI symptoms and autism spectrum disorder, you need to know how many patients he had to go through to get those 25.
Yeah, I reduced that down to just the guy saying I didn't cherry pick. I picked these consecutively, which is more or less randomly, but in a chronological order.
Well, you see, if a patient didn't meet the criteria,
sure,
they were out.
Yeah.
So it's selected. So they selected those who are showing the symptoms of both, and he's trying to draw a link between them, and he's missing out those, obviously, who don't meet the criteria, who don't show a link.
One of the things when I studied psychology was for an experiment to be scientific, it had to be it had to be replicatable, objective, and controlled. And this suggests the objective part may not be altogether objective.
Yeah, it's because it's so selected.
Well, I mean, the medical research always makes people roll their eyes when it comes to the subject selection. Because there's all sorts of niceties that you're taught in terms of when you do a true experimental design that you just can't do. I mean, they're just ideas that you give up on, like the idea of random assignment to groups. No, you are not allowed to take 100 Down syndrome of your patients. You can't take 100 of them that have Down syndrome and put 50 of them in this group and 50 of them in that control group and give them this drug and them this other drug. Consent is always a really big issue. But yeah, no, I sort of took what Concordance was saying from this thing too, that as opposed to saying, I mean, another way that he could have done it was if he had 100 patients that would have met this criteria, he could have put the 100 names into a hat and drew 25 of them.
Right. I mean, they have programs that replicate that, basically, that do random assignment. He could have talked about blinding. I didn't actually see any references to blinding anywhere in here.
Yeah.
I presume that wasn't important to him based on the arrays, but I don't know. That doesn't bother me so much. I'm used to seeing that kind of thing. That's why most researchers prefer mice, right? Because they're clonal, and you can control what they eat, and you can control what their drug exposures are. This is like splatters on the wall with all the different drugs and diets, and so a lot of these kids were getting this intravenous B12.
Yeah.
Right. They were getting MB12 vitamin shots, and probably by this gastroenterologist.
Yeah.
Well,
and
there's always a drive to. Oh, go ahead, Fiona, please.
Okay.
No. Becky was trying to say something there.
Okay, well, one thing that I discovered in doing my research is one of the reasons that a lot of these things haven't been narrowed down is because of the ethical considerations of doing studies on children. If there doesn't seem to be any positive effect to something you're doing, you don't do it. So a lot of these diets and, given certain medicines have not been studied, or the studies were stopped once they got started because of ethical concerns. So yeah, that leaves a big gaping hole, especially when the parents come forward and say, "But I've been giving my kid this drug that I got off the counter at the health food store, and the kid's a lot better. We need to test this drug." Well, you don't just randomly decide to test drugs because the parents say so. Although they did in one case, which I could hang on just a sec. I've got the paper up here. It goes from it's in Science. It's from 2001, where a woman said that it said the story of Victoria Beck is dramatic. Lorenzo's oil of autism. Beck's belief that the drug secretin radically improved her son's symptoms, and the media storm that resulted raised the hopes of many parents and galvanized public and private research. But the weight of evidence so far suggests that secretin doesn't work for the core symptoms of autism, at least for most children. And the National Institutes of Health did some quick studies and showed that nothing happened. There was no positive effect to it, and the fuss over secretin stopped. But everything I've seen here is people coming up with miracle cures of this sort of homeopathic remedy or this sort of herb or whatever, but they aren't necessarily going to be tested if there's no evidence that they're going to have any positive result, and if there's even the slightest evidence that they're going to hurt children. And it's like you said, Concordance, this is why scientists like mice.
Yeah.
And in fact, it is on IV secretin, a porcine version of, and also IVIG. So, pumping the kid full of, and this is a young girl, a 4-year-old girl, getting stuff on here that, I haven't seen this outside of a cancer scenario. So that's why it boggles the mind that you would even advance this as indicating really much of anything. Gene expression in kids under treatment with megadoses of flax oil and vitamin C and N-acetylcysteine, multivitamins of who knows what. There's actually 2 of them on here, the secretin. So yeah. It's a real problem for the study overall, and the study design itself is not very strong. And then I'll stop there and let Fiona set the pace.
I'm just thinking to myself, somebody's complaining that there aren't any Q&A. And I'm really sorry about that. Given the technical difficulties that I had trying to start this hangout, I completely forgot to enable Q&A. So if anybody's got questions, please post them in the YouTube feed. Cliff's keeping an eye on it. And so if any questions pop up, we'll try and deal with them. Now, the next bit that is interesting that I picked out. Cliff, if you go to 8 minutes 25 and screen share a still, so this is the handiest way to do it because it's certain parts of the paper that just absolutely leapt out at me. Right, now this bit made me think, wait a minute. Well, this is the diagnosis of autism spectrum disorder. And it gives you a breakdown. We've got 16 have been diagnosed with autism, 9 with autism spectrum disorder, from the following specialities. And if you look at them, we've not even got uniformity of diagnosis here. We have pediatric neurology, developmental pediatrics, pediatric psychiatry or psychology. And then if you go to, let's pick the time for the next.
I should, before you move off that one, one of the terms ought to be addressed because there's an experimental concern that comes from it. The NOS. Anytime you see a diagnosis referred to and then it's got something, something, or something, and then dash NOS, it means not otherwise specified. And it's meant to sort of be the garbage can diagnosis. It's just where all these other diagnoses fall. So, for example, he's saying diagnosis of ASD, so that's the autism spectrum disorder, and then parenthetically notes to be in this group with what we're calling a diagnosis of ASD, you would have as a diagnosis either autism or Asperger's or PDD, which I might need a reminder. That's persistent developmental delays or disorder, something. Anyway, I think it's persistent developmental disorder, where you either, not delayed, or because you can regress back, but it means not otherwise specified. So it means you had some persistent developmental delay or disorder that isn't attributable to some other condition.
Yeah.
Well, that's now, under this study design, that's now magically autism.
Yes.
ASD, not autism, ASD. So they were included. Basically, if the reason for their apparently persistent developmental delay had not been identified by the ripe age of 5 years old, they qualified for inclusion in the ASD group, which I'm baffled by.
Yeah, it is very confusing. So we don't even have any uniformity in the use of that specific criterion within the study. If we go into this next bit, Cliff, is at 8 minutes 59, if you could screen share it. And this brings it in a little bit.
That one?
No.
Sorry, that one.
It's 8:59, bang on. Yes, this is the one. Not all study participants were diagnosed with ASD using the same standardized diagnostic tools and by the same clinicians. And then I counted them all up. And I got to 7 different testing methods. But it also says among others. So I'm assuming it isn't just one other, there must be at least 2 others, which takes us to 9 methods. 9 different methods of diagnosing ASD in 25 children.
For 25, yeah.
And then it says after that, some patients were determined to have met the DSM-IV criteria without use of a quantitative score on standardized testing. So
this is digging now into the controlled element that I was saying, the rock.
I mean, how many of these 25 children truly had autism spectrum disorder?
Mm.
We can't really tell, because they only say some. And we know at least 9 testing methods were used.
And not only that, that rock thing I was saying about, the first one is replicatable. So how are you supposed to replicate this experiment to verify it under a peer review kind of system?
Yeah.
You know.
I have a real problem with that. That they've all been diagnosed with different methods. I mean, if we take off some patients were determined to have met DSM criteria without use, that must be greater than 1, so let's call it 2. So that takes us down to only 23 patients and 9 different methods. So we're now getting to the stage where there's only 2 and a bit patients have been tested by the same system.
Yeah, and even if you take the 23 or the 25, whatever number you want to use, and then there's 9 different methods, surely there's going to be some examples of participants that are in this group that would be the only one diagnosed with that method, that is just idiosyncratically determined to fit within that group.
Yeah.
I'm really, this part really puzzles me. Now that line, that some patients, without even providing a number of how many patients there were, it's hard. I try not to be too judgmental. Writing papers is hard, and you're always getting told, well, there's too much detail, cut these words out, and that sort of thing. But I just can't see how they can get away with saying some patients without so much as putting a number in parentheses. Actually, sorry, the normal stroke out on this part would be the word some would be replaced by number and a question mark after it. And you would be required to put down 4 patients were determined to have met DSM-4 criteria without use of, et cetera, et cetera. So the word some in that context, where a person could have used the same number of letters, you know, the same number of, like, the same amount of ink in the journal, so to speak.
Now, sadly, concordances had to drop just as we're getting to the actual methodology. I think what we'll do is I'll just leave out the dangers of microarray and qPCR. I'll just give a bit of a blab at that point. If anybody has ever done a thing called DDRT-PCR, it stands for Differential Display Reverse Transcriptase PCR, and you can use that to amplify up using messenger RNA as using the messenger RNA as the template, and that would mean that what you'll get is a pattern showing which genes were being expressed at any given time. Okay? Now, if anybody's ever tried to do DDRT-PCR, they will know that it is a pig of a method. It's an absolute nightmare.
What is it?
It's where, when a gene is being expressed, messenger RNA is being transcribed from the gene. Now, you take your tissue sample and you extract all of the messenger RNA. And you then use PCR to amplify up all those messages. And it's reverse transcriptase because you are using the messenger RNA template to make double-stranded DNA. And you can select them all by size. Now, in that one tube, you're amplifying up all of the messages and then separating them out and looking at the pattern. And you can run 2 patterns side by side and see if there are any differences between the 2. And as I say, it's an absolute pig of a method. Because the sensitivity in visualizing it is a big problem.
It might help because I know there's lots of different forms of PCR, and I know of none of them or how any of them work. I remember learning about, PCR. Here's polymerase chain reaction. This is what you do to amplify a sample. So can I just dial it back a little bit and then correct me if this doesn't sort of get us to why that's a difficulty? Is that the rubber meets the road here of why anybody does PCR to start with. So as I was always taught it was like it's a nice way of doing a test for some sequence, right? Mm-hmm. So for example, Cliff, if you were concerned that you had HIV virus in your body, right? We could draw a sample of fluid from your veins or whatever, get the sample of blood or whatever, and hold it up and look at it and say, well, do you see any? Well, how would we find the virus? Well, let's look for its DNA. And we could look for it and we wouldn't see it. We could take a magnifying glass, right? I don't see it. Well, just because we don't see it in your blood doesn't mean you don't have it, right? We could look at it under a microscope. We're still not going to see it. Well, we could do a test for the chemical properties of that DNA. We're still not going to find it because it would occur in such small trace amounts. So apparently, as I understand it, very limited, but the PCR is a way of saying, well, what we can do is if there's some of that DNA that's in there, we can cause it to replicate through cycles. That's why it's called the chain reaction, the polymerase chain reaction. So it takes a section of that polymer, makes another copy, and it's got something to do with heating and cooling and some reagents being present. I don't know. It goes through cycles and cycles. Every time it goes through a cycle, it does a doubling. So you put it through a cycle, put it through a cycle, put it through a cycle. So you take that bit of virus DNA that's in there, and you double it, and you double that, and double that, and double that, and double that. Well, pretty soon now you've got a detectable amount of that DNA. So you can look and say, hey, yeah, bad news, Cliff. Turns out you do have HIV. I don't know why I chose that as an example. I should apologize. You do have HIV DNA in your blood sample. But imagine the circumstances where you didn't have it to start with. No amount of doubling would actually lead to detectable amounts because it's double nothing, double nothing, double nothing, double nothing. So the reason I present that is completely unrelated to what we're talking about. It's just to sort of ground us on why do we do PCR anyway. And my understanding is the reason we do it is because we're trying to answer a question, almost a yes or no question, about the presence or absence of something. In the case of virus screening, you're trying to ask a yes or no question: does this person have some trace amounts of this DNA in their blood? But I think I'm like, that's so remedial and elementary to compare to what Fiona's talking about, because now you're talking about you do amplifications of, instead of the DNA, you're doing amplifications of RNA. And not just one time. Yeah. Okay. Yeah. So now, knowing that that's what the test is for, can you explain that again? Because then maybe I'll catch it the second time.
Right. Okay. I'll sort of explain the difficulties.
Can I just?
Right. One of the problems
is this a bit like, remember when we looked at the Andrew Wakefield one, and you had where it was replicated and it went up, and the threshold was adjusted.
Yeah. No, it's not like that. No, it's not like that. It's trying to determine what has gone over a threshold. That's the problem. But yeah, it's the same sort of methodology. You amplify up the target.
Okay, multiplication of it.
Yeah. Your problem comes with when you're doing something where you're trying to assess if a gene is being expressed or not, and you're looking for this difference in the pattern of expression. You don't have any specific genes that you're targeting. So what you have to do is use random primers. To do PCR, you need 2 primers, an upstream and a downstream, right? And every message in the eukaryotes ends with a poly A tail. So it's a tail of adenines right along it. So knowing that downstream primer you can make as a row of T's, and it will bind. So at least you've got one primer that you can use. But for the other primer, given that you're looking at a whole population and you want to amplify up all the differences, you have to use random primers, little hexamers, 6 bases or maybe 8 bases. And as long as you use that same one in all of your samples, if your samples are identical, you should get an identical profile. And if there is a difference in the gene expression in your samples, you should see that difference. It's not guaranteed. the big problem is that it's not one of the most reproducible methods out there. For all sorts of reasons, and one of them is because messenger RNA is a bastard to work with. It's horrendous. You look at it the wrong way and it degrades. And that's because there are enzymes around called RNases. Okay? They're produced by organisms. You find them in the cytoplasm of just about everything. And their job is to degrade messenger RNA because it's one of the methods of regulating gene expression. If the message hangs about in the cell, it would just keep being used to make more protein. How would you control it? And so there are these enzymes which chomp up the message. So if the cell wants more, it needs to make a new piece of messenger RNA. That's a way of controlling it. Now, these enzymes are very resistant. They resist autoclaving at 121 degrees and 15 pounds pressure for 15 minutes. They're an absolute nightmare. And so anybody who works with RNA has got a lot to swear about. Because no matter what you do on a given day, when you have your lab coat buttoned up to the neck, your hair soaked and slicked back, 3 pairs of gloves on, and you've told everybody on the benches round about you to bugger off and leave you alone, you can bet your bottom dollar that sitting on your bench right beside you is an RNase molecule with your name on it. And you don't know that until you actually look at the RNA that you've isolated and see if it's degraded or not. Mm-hmm. It's horrible working with RNA. And just as an aside, I isolated intact RNA on a trawler at sea in storms. I think I'm the only person stupid enough to try it. But it's not easy and it's nasty chemicals. Now, Concordance wanted to start saying about the difficulties in the specific methods that were used here with the microarrays and qPCR. So off you go. The floor is yours. I've done what I can.
Fiona, did you notice that they used GAPDH as a housekeeping gene? Do you see any problems with that in a gastrointestinal concept?
Before you start, is there anything you want me to screen share to, if you need to illustrate the point?
No. I'll hand wave behind my icon here. The reason why that's funny is because you're putting a little bit more of one sample and a little less of another. You have to make sure that that doesn't look like, oh, there's a whole lot more of this message. This gene switched on a whole lot more, when in fact you just added a little bit more of that sample. So you have these reference genes, which are supposed to be genes that are always turned on at a very low level. And so you say, well, there should be, about 100 copies for every microliter, every tiny little drop of the sample. And if you measure a little bit more or a little bit less of this from one sample to the other, you say, I put a little bit more of that sample in, I put a little bit less of that sample in based on where those are. But the genes that you pick are one of the biggest weaknesses in how the data is analyzed. So in this case, they've chosen something from the Krebs cycle, from normal, oxidative phospho, just normal metabolism, okay? And in this case, we know for a fact that these cells upregulate and downregulate, that these genes are switched on and switched off because this is the kind of cell that is required to have these metabolic processes modulated based on what context they're in. We know people with diabetes, for example. You really can't use this gene because it is regulated based on the disease state. Likewise, they use actin. Actin is like the skeleton of your cell. They're one of the many components that gives the cell rigidity. So if you have a cell that's inflamed, we know for a fact that actin may go up or may go down. So when you're normalizing to these things, you can actually create, by trying to compensate for your own sloppiness, you can create data. And it's one of maybe the biggest weaknesses of the whole technique, qPCR, real-time PCR. And what they've got essentially in this paper are two techniques that are great for identifying targets, possibilities. You know, it creates a lot of interesting questions to be answered by additional confirmation. But that additional confirmation is not being done here. So they say it in their abstract. And one of the last lines in the abstract is, hey, don't take all of this with a grain of salt, because we know that we need a validation population to know whether or not to even believe this data. And to a scientist reading this, they're saying, pardon my language, but no shit. Yeah, these are preliminary, highly speculative types of experiments. And yet, I think it could be presented in the popular media, and if I were a more cynical person, I might even say this paper was designed in such a way that it could be presented to people who already support the Wakefield hypothesis linking gut inflammation with autism status. It could be presented with all its methods to a lay audience to be persuasive that perhaps mutation is causative. When any scientist reading this, you've done a couple of gene expressions, you've measured some numbers here, but where's the follow-up? Where's the Western blot team that's supposed to be made by these messengers coming from the genome out to the rest of the cell, where they're supposed to be turned into something that has an actual effect? It's a bit like saying, I don't know if the light is on, but when I stick my finger in the electrical socket, I get electrocuted. Well, yeah, that may mean the electricity is working somewhere in the house, but you don't know necessarily that you're actually turning on the light you think you're turning on. So they're confirming one very suspicious technique with another even more suspicious technique, and calling it a day. And I think any researcher worth their salt is looking at this with a very suspect eye. Fiona, I'll pass that back to you there.
Yeah, it is very interesting. I mean, I think if anybody has done work with trying to look at gene expression levels, they will understand exactly what I was getting at with the difficulties that there are in the method.
I did see one very interesting point where they compared the microarray results. This is where they're trying to get a match between a sequence and a messenger RNA from the cells. And there's a mismatch to what they got with that second technique, which is the qPCR, the quantitative or real-time PCR analysis. They generally pointed in the same direction, but with very different magnitudes. In other words, if one technique said this gene is switched on, it's switched on by about 4 times as much as it would have been in the healthy control. Then the real time also said it was switched on, but that it was switched on 400-fold. So there's some pretty big differences in the magnitude of change detected by the two techniques, to the point where it really makes me suspect that they're not doing proper controls and they don't understand this technique well.
Yeah.
If I can toss in a clarifying comment, I heard you mention probably about a minute and a half ago the Western blot follow-up team. Do I recall correctly? Western blot actually tests for the presence of proteins, not for nucleotide sequences, right?
That's right.
Yeah. So I think people's recollection from the nice debate we have with creationists on YouTube is that if DNA codes for proteins, you can have DNA, but so what? Those sequences are there for whatever purpose. And I don't think that DNA really has much of an effect on the body. It's when it's encoded into proteins, those proteins tend to have an effect. And if we say, well, it turns out there's this interesting sequence, nucleotide sequence in amongst the DNA, then does that turn into there being some, potentially difficult or problematic proteins present? And there doesn't seem to be a suggestion or a citation of a follow-up article, that is a follow-up article citing this article saying, yeah, well, hey, we found this, and guess what? It turns out we did some Western blot and found out that the protein in these subjects, but not those subjects, this particular protein is present. And I don't think there was such a follow-up.
No, but it's interesting to me. They did sort of an internal follow-up. They used the microarrays, and microarrays are great because you can screen 20,000 human genes. Are they turned on, or at least at the transcriptional level, at the level of making a messenger that's supposed to go out and make a protein? You can verify that they're switched on or switched off. What struck me as a little odd is the number of genes. Fiona, I don't know if you noticed this. 178 differentially expressed transcripts. That's a lot.
Mm-hmm.
I would expect those kinds of numbers between like a really serious pathology.
Mm-hmm.
Someone with Ebola might have 200 genes switched on. So 180 genes differentially expressed is too high a number, which suggests either they've got something else going on or they've set their threshold way too low.
Yeah, or there is the usual problem with reproducibility when it comes to differential display. It really is an absolute pain in the neck. I've attempted this in 2 ways. In my PhD, I was doing, for my sins, 2-dimensional polyacrylamide gel electrophoresis, which meant separating the proteins in 1 dimension on their charge, and then in the 2nd dimension on their size, and getting a pattern response. And you were looking at, in my case, starving bacteria and non-starving bacteria, and looking for a difference in the proteins. And even using the same sample and running the gels twice, you couldn't even guarantee you would get the same result on some of the proteins that were maybe just barely present in one and not present in the other. You know, it was the real in-your-face differences. A huge big blob for, it was OMP-A that I finally homed in on. That one always turned up, but in some of the proteins that there was less of, you couldn't get a reproducible result. And it's all down to the various limitations and difficulties of some of these techniques. I don't know if you've ever tried 2-dimensional SDS page, but it's horrible. You're working with 9 molar urea that can crystallize in your polyacrylamide when you're doing the 1st dimension. So you have to run it in a warm room at 37 degrees. So you stand in that room for 6 hours. Absolutely unbelievable how difficult it can be to get reproducible results from that, just looking at the proteins. But when you're actually working with something like messenger RNA that has this additional problem of degradation from one sample to the next. And also remember that the tissue sample that was taken, the cells in that sample contain RNA's enzymes that will break down RNA. And when you take tissue samples for RNA, you've got to take it and drop it straight into liquid nitrogen. And it's got to be small enough that it snap freezes to the center. Any delay, and you will start getting degradation of any message that's there. So there is a problem using techniques of detecting gene expression in tissue samples, especially when they have a child in theatre.
Yeah, I was going to ask, how does that work? Because I was mentioning the tools before for the excision of samples for biopsy. You normally have a liquid solution. So you pull the loop or the little toothy thing back out, and you say, and it goes bloop into a little jar, and then there's usually an assistant there that will turn the lid on the jar, but that's intentionally a liquid that it's not meant to be stasis in any way. It doesn't actually freeze it in any way. I mean, are you suggesting that to do it properly, you'd have to have a bucket of like liquid nitrogen in the room, like?
Let's go back to the paper here. In accordance with routine diagnostic biopsy protocol, immediately upon procurement of biopsy tissue, a specimen from each of 7 anatomic locations was processed for paraffin embedding and subsequent routine histopathology. Biopsies for microarray analysis were obtained from the divided mucosal specimens at each anatomic location. These tissues were placed directly into RNA later, which has an RNase inhibitor, and stored at -80 prior to processing. So there's a preservative that you can dunk them in that will keep them alive a little bit longer, but there's always degradation. It's like a ticking clock. The quality of the RNA drops from the moment you have it in your hand.
Yeah, the only way to really be sure is liquid nitrogen. And then if you need to ship your specimens anywhere, they're shipped on dry ice. So that's solid carbon dioxide. That's the only way to be sure. Freeze that it's absolutely solid, so that there is no way that an RNase enzyme can do anything, because there's no liquid for it to function in. I mean, as I say, I've been doing RNA isolation, etc., for quite a while, and it's not an easy technique. And you can just imagine messing about with phenol and liquid nitrogen on a trawler in a storm. It's not easy. Even within a laboratory setting, trying to get samples from dry ice to liquid nitrogen, back to dry ice, to, when I was doing it, you had to do the extractions in a fume hood, which is not a clean environment at all, because that's dragging air in. You've got all sorts of problems with that. And you have to do that because of it was the phenol in the RNA isolation solution that I was using. The best one to use at the time had a lot of phenol in it, and it was really nasty. The more dangerous it is, the better it is at isolating RNA. So that does make you wonder about each sample. I mean, how uniform was the storage? It's not easy in a theatre environment when you're taking one sample from a certain location for histology, and then you need another sample from the same location for the RNA analysis. Or do you take one sample and divide it? That would be the way to do it.
So I can say from the paper that they said, and Fiona, I don't know if you've ever used an Agilent bioanalyzer, but they set a RIN number of 7, which is it's a technique designed to separate all the little bits of RNA, and the smaller the little bits get, the more chewed up the RNAs become, and therefore useless. Once it's become chewed up, we can't test it anymore. Yeah. And that RIN is a way of expressing quality. So a RIN of 7 is pretty modest, which to me that they were willing to accept some pretty, but not out of range. I'm probably cut and pasted from the manufacturer. Make sure all samples have a RIN of at least 7. So what bothers me about this, and this I will promise not to go off on a diatribe about this, but the problem with this kind of data is that the longer we have these techniques, the less people feel the need to describe them in detail. But the details make all the difference. So as a result of Wakefield's work in this field, we've actually instituted a new standard called the MIQE, or I call it MIQE because it reminds me of the Life cereal commercials. The MIQE, it's the minimum information necessary to publish quantitative PCR experiments. They skipped a bunch of letters along the way, but the point is it's a stand for publishing data that requires that researchers be explicit in how they have handled the samples, what the mass of the sample was, how long it sat on the bench, how long it sat in a very, very cold freeze, how was it processed, what kits were used in the process of obtaining the sample, when it was run, how did you design the experiment, where did those assays come from, how were they tested to make sure they're legitimate and going to produce quality data. This paper doesn't mention anything about the MIKE type standards. It's also microarrays. It's the Miami standard, which is MIAME, minimum information to analyze microarray experiments. None of those were given here, and a lot of it being skipped over. What they've done, I notice, is bought everything as a pre-made shake and bake type kit. Bake translates well to the UK, but it's the idea of something where you plop the chicken in the bag and you shake it with the crumbs, and so you've got a coating for your fried chicken. You put the things in the container all in one, and you mix it all together and you run it. These are very low complexity types of experiments. I made the comment, I think you could probably give this to a grad student to do, and it would probably be done in 6 months or less. It was a remarkably low complexity study based on the techniques, which makes me think, combined with the fact that none of these people have published on this type of work before, that this may have been the first time they're doing it. And when reading that just heightened my skepticism again. If these are not transcriptomics people who routinely use these techniques, they're not aware of all the pitfalls that someone with more experience would recognize, like choosing GAPDH as a housekeeping gene, which was, I had to read it 3 times to make sure I'd gotten it right. That's a terrible choice for them to have gone that direction, and probably invalidates the results. The other question that comes up as I read this is, how did this pass peer review? What referees read this and said, yeah, absolutely, totally, they had perfect paper, send it on? What do you think, Fiona?
Well, it's
Andrew Wakefield.
Yeah, it's public.
Maybe he was on the review committee.
It's Public Library of Science. PLOS One.
Right.
It was published in. I'm not sure of the review standards of that. I need to look into that a bit more.
Yeah, PLOS One seems to have a mixture of. PLOS One has come, I think, real mixture of high quality, bold studies and kind of sloppy research too. There's no limitations on how many papers you can throw out in a month. It's just how many you can get reviewed.
Yeah.
Yeah. What are their different sections? This one's in the research article section. I mean, there are some journals that are broken down into different things with different standards, right? So there's like, even the New England Journal of Medicine, which people would say, oh, well, that's a pretty prestigious thing. Well, they have a letters section that has about the same scrutiny as the letters to the editor of your local newspaper.
Yeah.
I think that's unfair, but it's not peer reviewed, is what I'm getting at. So research article. Do we know what the it says? Peer reviewed research article.
Let's have a look at something. Let's just have a look.
Well,
I was just looking at their Cs, and they tout the fact that every article is reviewed by at least 2 peer. It says all accepted articles have been reviewed by 2.9 experts, which I don't know quite how that works.
The third guy,
I guess one guy's really short.
It's current impact factor. Well, go away. I got a pop-up. Its 2013-2014 impact factor was 3.534. So.
Yeah, they charge.
How do you get 2 points?
Over $1,000.
Is it one review that's just not all there?
It's interesting. So I don't know how you get more than. It's one academic and 1.9 reviewers. Data refers to the manuscripts submitted. Yeah, external peer reviewers. Somebody missed.
Somebody's breaking up.
It went a little bit tinny there, Concordance. So you might want to try that again. What did you just say?
I find someone without a limb. Someone missing a couple fingers.
Oh, you're still on the 2.9. Yeah. The impact factor sounds a little higher to me than what I expected to be. And sorry, in the instructions that you pay to get stuff published in it. Well, I guess they're non-profit,
yeah.
but
one of the more expensive. It's a product of the Library of Science, right? Public Library of Science.
Yeah.
Is that what it stands for?
Yeah, and it's. I mean, there's good stuff on it, but like everything, there's good and there's bad.
Well, I wonder, can we, if you want to just wave this off if this takes us off topic, but what is the purpose of peer review? I know Concordance and I have been off and on for more than a year now saying we should do a segment on Magic Sandwich Show about the intended effect of peer review and the actual effects of peer review, and then we never get around to it. But I think because we're now sitting here, I mean, we're initially discussing some fairly detailed stuff about laboratory procedures and whatnot, but now we're kind of looking at the article itself and how does this come to be, and it says peer reviewed, but what is the purpose of peer review? How should knowing that an article has been peer reviewed change our expectations?
Well, a peer reviewer will do exactly what we're sitting doing here now, looking at it and saying, what? They're looking for any flaws in the methodology that may have an effect on the results and the conclusions. It's very easy, and in fact, maybe we'll do another hangout on another paper, which is a beautiful example of setting out to answer one question, and by the way you've designed the experiment, it gives an answer to a completely different question, but you then use it to answer your first question. So we'll maybe do that one. That's a beauty. That's the sort of thing that the peer review process is looking at. And so the things that Concordance and I have been highlighting about difficulties in these techniques, and not having full information about the samples, the actual patients. These would all be flagged up by a peer reviewer. What peer review can do is they can recommend a paper for publication. They can recommend it for publication with some changes, and they can recommend what changes you should make. Or they can recommend not publishing. You need to do further work, and they can actually recommend what work you need to do to close those gaps that they've found. Because one of the problems when you work on a piece of research for any length of time is that you can end up with tunnel vision, and you don't see some of the more extraneous implications. And a peer reviewer coming to it as a fresh mind reads it, and they don't have that tunnel vision that you have. And they see all these other factors. And peer review is a way of making sure that the actual work that was done in looking at the materials and methods is actually up to standard. That they haven't done anything really stupid. And I mean, there have been papers rejected because the flaw in the method used has been so horrendous that there is no way it can be published because they've done it wrong. They've actually done their experiments wrong.
The protocol for usually how peer review works is that you send it to anonymous reviewers, right? Like the journal editor will just send out the manuscript, like the original paper, to maybe 3-4 random, researchers in similar fields to that the author or authors are in who've written the original paper.
Yeah, in fact, you can volunteer names of good peer reviewers for your own work. which I think is a system that is prone to abuse. There have been cases where researchers actually created fake identities and recommended themselves to review their own papers.
Oh man,
there's some other pretty bad cases of fraud, but I think scientific fraud is kind of on a low end right now. I think it's only about 1 or 2% of papers really are caught with fraud.
Yeah, and I'd like to emphasise we're not suggesting there's any fraud in this paper.
I don't think it even needs to.
I think I'm highlighting the flaws in the peer review system, which I think is pretty badly broken.
Yeah, I just wanted to make sure that was said so that nobody could accuse me of having said that.
Yeah. No, that's fair. That's fair, and I think that we even notice in reading this paper that there are places where they give caveat. They say, well, this is we use this. This is what we found, and it doesn't necessarily mean that. It suggests this. They were quite cautious themselves, and the danger of releasing a paper like this is the fact that the popular media or politically invested individuals can take snippets out of it and turn it into something that it's not. So I don't get the vibe that there's any sort of fraud here at all. In any case, what I find weird about is that the peer review process, even when it functions correctly, only looks for certain things. And I think that the account we're giving here really focuses on, as a referee, what you receive is a copy of the manuscript. They don't send you the data on a CD or an array of photographs and ask you to sort of repeat their investigation and their analysis. They send you the manuscript, and you decide based on the background provided in the paper and the methods as described and the specific results that come up. Is the discussion section, what are the implications of this research? Is it warranted from the information provided? And the most common reason that these things get bounced is like, wow, your background was insufficient. You didn't list this and this and this and this, and then your conclusions you draw from this thing in your discussion section is just crazy to the moon stuff. It's not actually supported by the thing that you've done here. And then, of course, when it's not a rejection but to sort of revise, resubmit, it's because they'll take exception to certain methodological things. I have to say, I really struggled with this paper. You shown it to me in the order of a month ago, 2-3 weeks ago, and I'm not familiar enough with the methodology that's going on here. And as I listen to you and Concordance describe, things like, oh, can you believe they use this as a reference marker or a reference sample? Like, oh, that's crazy. And honestly, I don't know. There are things I certainly like the use of principal component analysis as a statistical technique to then be able to make this scatter plot that shows, look, we've got some colored blobs here and some colored blobs here, and therefore that illustrates an important difference between these groups. To me, that really stands out as that's not the right analysis for what you were trying to say here. And that's the kind of thing that peer review gets into, where the referees, if they're familiar with some little subset of the thing you were doing, they're going to be able to point to those things. But they certainly don't look for fraud or reanalyze your data. And the most they can do is send back questions. Like, for example, the one I was pointing out earlier, when somebody says, well, some of the subjects were selected even though they didn't actually have numerical testing that was done. It was sort of done because we felt they met the criteria. Well, you'd cross that out and put in there, well, how many of them? We need you to put in the specific number in this place, because that's just good scientific writing. But the idea that somehow the actual data are reviewed and that they make sure that the results are correct, that never happens in peer review. But anyway, having said that, I still think that this paper, if it was sent to somebody with my level of knowledge of how PCR works, that referee should have said, I'm not qualified to review this paper. And if it was sent to somebody with, like the Concordance goes into these details about it, I would imagine, Concordance, I don't want to speak for you, but I would imagine you would have said, no, can you do the analysis properly? I don't know. What would you have said?
Yeah, I think the other thing I just wanted to highlight, and then I'm going to go to...
By the way, real quick, the PCA analysis, the cool blobby-looking charts, are directly from the software that they used. I would have expected them to sort of regraph it into something a little bit more neutral. And PCA in 3 dimensions on a 2-dimensional sheet I think that's a cardinal sin too.
Yeah.
You can't tell where those blobs are aligned in the third dimension because they've got the rotation wrong.
Yeah.
And I will correct you on one other thing. Sometimes, if the researcher is complying with transparency standards, they will be required to submit their raw data. And again, this is a new thing. These new publication standards require that they are submitting in these generic formats, right? Because each of these instruments has a manufacturer with specific software that will only open their files. They're required to submit the data in a form that a referee, that a peer reviewer, could take that data and reanalyze it to make sure that the numbers actually add up. Unfortunately, the Wakefield paper, and I've made a video on this on YouTube, the Wakefield paper did not comply with those standards, and in fact, when they went back to the raw data, they found evidence of manipulation of the threshold. This is why he was struck off, one of many reasons why he was struck off. Manipulation of the thresholds to produce the desired result. For me, that is absolutely the smoking gun. There's no way you did what happened there. That could not have happened by accident or even sort of indiscretion or lack of judgment. This was an intentional act.
Yeah, I mean, if you've ever done real-time PCR, when you look at those plots, his plots are terrible.
You mean the plots in this paper?
No,
yeah, they failed. They failed, but he's got them on log scale, so they look like basically he was reading noise and calling it positive amplification of measles virus.
Yeah.
And it certainly wasn't.
And
if you've ever done...
his standards met one threshold, and his samples met another, because if they had been on the same standard, and I did them side by side, there was no measles replication in any of the samples that he called positive.
Yeah.
It was not an accident.
If you've ever done real-time PCR, I mean, if a student had come to me with that result, and I knew it was a positive sample, they would get a cuff in the back of the head because all the tests were negative. They were negative. They were not positive. There was no exponential increase, and you've got to have that exponential increase to show that there is a chain reaction happening. That's the whole point of the polymerase chain reaction. He had none of that. They were negative. They were cast iron negatives. There's not even a quibble about them. Absolutely negative. It's not just one of these things, oh, well, somebody might think it's, no.
Sorry, that's going back, that's referring to the Wakefield.
Yes.
Wakefield. Yeah.
And I mean, the threshold is set automatically by the real-time PCR machine, right? And what it does is it looks for the exponential increase in your controls, and it puts the threshold at the halfway mark on that. It does it automatically. So for the threshold to have been lowered as much, you had to go in and manually change it, because the machine would never set that as a threshold. Never.
This concordance, I had not heard of this business where. So when you're saying there's a standard now when you're reporting these results that you would actually provide some consumable data to the referees, is this a thing that's specific for this testing, for this technology?
Yeah, but unfortunately, it's only certain journals that have adopted this standard.
Okay.
And honestly, people push back against it so hard because the reviewers don't have time. It sounds noble and lovely. Let's replicate someone else's results using their raw data, but referees are unpaid. They're taking time away from their own work to dig through the data of someone else, potentially a competitor. And their only motivation is to find something they can spike them with, perhaps, but they really just don't have time to do the amount of work required, so they don't want the data. They honestly are not real enthused about it, and they don't plan on doing anything about it. It's a nice thing, but it's not likely to have the desired effect. Fiona, I don't know if you have a different opinion.
Wasn't there, after the Wakefield fiasco with the real-time PCR, wasn't there a rule, but in some journals, where the person who actually carried out the RT-PCR had to be specifically named?
Yes, not only that, but where the samples, who handled the samples and how long they sat in the freezer.
Yeah.
And those are big questions, never answered in papers. There's kind of details you think, well, no one's really interested. That's just going to take up space, right? Mm-hmm. We've got a page charge to worry about. Let's cut that down.
Mm-hmm.
But I want to know. I want to know if you're comparing samples from 12 years ago to samples just freshly harvested. That's going to create its own effect. And particularly if you have, let's say, a long list of disease status patients, and you're collecting the samples over a period of years, and then you enroll controls, meaning you've got this sample spaced out over 6 or 7 or 8 or 9, 10 years, and all your controls are fresh. Well, yeah, you're going to find a difference between the control and the treated, and it's going to be a result of 8 years of sitting in a freezer, right? You might imagine if you took your frozen food and you compared it to fresh food, that the tastes are going to be different. It's exactly the same scenario. And that's just not published. They just say 25 people were recruited. 25 consecutive people who met the criteria were recruited.
Yeah.
Right? Those kinds of descriptions are not telling us important details about the experiment.
Yeah. And I wanted, now that we're talking about controls, I've given Cliff a timestamp for a wee share.
Yep.
And this is the one. This is the sort of the headshot. This one blew my mind. I was looking for this information. Yeah, this is the one. Right, now, I wondered right from the outset how they managed to get permission from an ethics committee to be taking biopsies from children without autism and without bowel disease, because we know that that is one of the things that spiked Wakefield's career.
And not just shoving the tool up their bums, but taking chunks of tissue, very small chunks of tissue, from 9 locations in their gut. That has a risk associated with it.
Yes, and I mean, I think one child in the Wakefield study, he had his bowel perforated multiple times, and he almost died. But anyway, so I was looking for this information, okay, and I came across it, and this blew my mind. Prospective controls recruited through an IRB-approved protocol, blah, 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 blah. I'm looking for the end of that. From the Paediatric Gastroenterology Clinic at the Wake Forest University Health Sciences. Included children who presented with symptoms and laboratory testing suggesting possible intestinal disease. Crohn's disease, ulcerative colitis, celiac disease. So that then answers the question how they got ethical approval for this, that there actually was a reason to do the biopsies. Then it says, non-IBD controlled subjects were further defined as those who, following colonoscopy, were without endoscopic or pathologic findings explaining their symptoms. However, the initial indication for colonoscopy was presence of unexplained GI symptoms ranging from abdominal pain, diarrhea, malnutrition, and blood observed in the stools, etc. There is no way on God's green earth that you can call them a non-bowel disease group. They have got symptoms of some form of GI disruption. How are they the negative controls?
Yeah, that part baffled me. I mean, they're increasingly it's not as difficult as traditionally one might think it is to get endoscopy, colonoscopy, endoscopy, even esophageal biopsy, on children. In part because the greatest risk to those things let's stick with the bowel exam, is the perforation. But the monitoring, like, I mean, the rate of disease from that sort of thing has gone down so much. It's actually considered to be less risky, so it's not so hard to do it. But you can't do it for research reasons. You have to have a physician sign on to say, like, yeah, this particular child for the symptom presentation needs this done. So I was sort of really puzzled by this part. It just seemed like they were saying that, well, we have some that weren't diagnosed with these conditions but still had other problems because, after all, we couldn't get symptom-free children to get biopsies from. So that's why we called this the control group. I don't know. I thought initially I'd misunderstood it. Did others get that impression as well, or?
Yeah, no, you haven't misunderstood it. So basically, the non-bowel disease control group has some form of bowel disease.
Do you think, I mean, I'm just playing devil's advocate here, do you think perhaps they have typoed, and what they mean is, yes, they've got bowel issues, but they are not showing ASD signs?
No, because remember, only one group has ASD. That's a test group.
Yes.
We have ASD and bowel disease. Then there's the non-ASD with Crohn's disease. There's the non-ASD with ulcerative colitis, and there's the non-ASD non-bowel disease control group.
Which has bowel disease or issues.
Yeah.
I wonder if they're I wonder if they could be the what was that one you said, Hogtie? The non specific one, and NCO, was it?
No, that was to do with the diagnosis of autism.
Yeah, NOS, not otherwise specified.
Yeah, that was in the autistic group. That was specified.
Yeah, that was when they were slotting who's autistic and who isn't autistic. They included amongst the autistic kids anybody who had a persistent developmental problem that wasn't specified as, like, you know, the kid had a head injury or something like that.
Yeah.
In there.
In
this case, so it does seem to be that they have. I'm assuming from the slide here that where they use the term initial indication for colonoscopy, that they're using the term indication for colonoscopy under the convention, an indication for colonoscopy meaning a good idea to do a colonoscopy, that it's not just it might be a good idea, but that a physician has actually endorsed the use of colonoscopy as something beneficial for that patient. So here you have what they're saying, the initial indication for colonoscopy, that is where it was flagged as, yes, it's a good idea this child gets a colonoscopy, was the presence of unexplained or not otherwise explained GI symptoms, ranging from, and then they list several different things. And I would have assumed in there they would have included food allergy, maybe food allergies included in the, etc. And then they say failure to diagnose the etiology of the observed symptoms by endoscopy was subsequently followed by clinical reassessment and additional diagnostic testing. So it sounded like they said, well, as long as we did the colonoscopy, or the was it colonoscopy or endoscopy? Colonoscopy. Then and then didn't find a diagnosis, then they were included in this thing.
Yeah, but you see, that's supposed to be the control group. They should be showing the non-disease gene expression profile.
Yeah, I would. By the way, I would not call that a gastric or colon disease group, because again, the disease should be used to mean confirmed disease, like where there's actually a condition, a diagnosis that's been dropped on the kid. Unexplained symptomology. I mean, they're certainly not declared disease-free because they have symptomology they presented with.
Yeah.
They are free of the disease label, was sort of the most that I could read out of those two sentences.
I mean, if you want to use something as a negative control, it's got to be negative.
But you see, here's where you come into the ethical considerations again, because no one's going to allow you to grab a bunch of perfectly normal, healthy kids and do a colonoscopy on them.
Well,
it's interesting because
with that in mind, when did they get these?
For the control, it's actually much longer than the medication list for, say, the Crohn's disease kids. Some of these kids were on 8 or 9 different drugs. Some of them, very mild. They were almost all on a laxative. At least half of them here look to be on laxatives.
That would be an
hydrocodone. I thought that was a little strange.
This would be an
and when were these samples collected? There isn't a chance they were dipping back into old data to find these kids
No
back
idea.
before they got caught doing these things?
I honestly don't know. I could not get past my head desk when I read that bit.
Okay. Well, I don't want to throw that out there as an unfounded accusation. I'm just asking if anybody else had noticed a date in there, because remember that was part of the problem with the Wakefield group to start with, was doing some of these things with sort of sketchy parental consent, and they may have had access to that data still. And I don't want to throw that out as an accusation, but I'm just wondering if there was an express statement in there that, well, this was data that was collected in, say, 2012, well, then obviously, no, that's something different.
No, I didn't notice that, I must admit. I'm not saying it's not there, but I didn't notice it. I just couldn't get past this. That if you've got abdominal pain, diarrhea, malnutrition, and blood in your stools, something's not quite right. It doesn't take a rocket scientist to work that one out. And so to call...
I know this might sound a bit of a weird thing. Does anybody remember where Andrew Wakefield went? Because at the moment, hasn't he got something
Texas.
in one of the southern states, Texas? Is that where he's staying? Because I'm just looking at where Wakefield University is, and it's North Carolina.
Wake Forest, not Wakefield.
Yeah, Wakefield, yeah.
Sorry, Wake Forest. Yeah, sorry, yeah. Wake Forest. Sorry.
No, Wakefield is at
I know.
In Texas.
It's like I say, I'm looking at Wakefield Forest University, and it's in North Carolina. And I know that he has been hiding out in America in one of the southern states, and I just wondered.
It was Texas. It was Texas.
He's about an hour from where I live.
Yeah, he was trying to use the Texas long arm law against Brian Deere. It was thrown out.
Yes.
When we talked to Brian Deere, Cliff, he was still waiting for that case.
Yep.
Remember? Mm-hmm. It was thrown out. I got a message in from Brian Deere about that.
Yeah, I was someone myself.
Yeah. So that's it. Wakefield's now had it, trying to sue the journalist. Now, this is a good point, I think, to drag Becky into this. I asked Becky to do the dietary bit for me, and it's all to do with the big spreadsheet that's provided as one of the supplements, and that's an eye-opener as well. And this has been brought up a number of times in the Hangout, but I think this is now Becky's turn. Off you go, Becky. You have the floor.
Okay.
Go for it, Lassie.
I have the floor. Well, because when we were looking at this, first thing I did was open the spreadsheets because I love spreadsheets. I love data. I love spreadsheets. Give me a spreadsheet, and I'm really happy. Give me data, and I'll start graphing it. What?
But you're weird.
Yes, I'm just a little weird. Give me data, and I'll start graphing it and analyzing it just because I find that entertaining.
Do you have SPSS or any other like statistical software for a statistical analysis?
No, I mostly just use Excel. I'm a master at Excel. I've used it at work for 20-some years, and I have other software I can use, but I usually get what I need from Excel. But anyway, what I looked at is I looked at these two spreadsheets, and I don't know if you can open them, Cliff. If you can't, I should be able to.
Yeah.
And, okay. It's if you open the first spreadsheet.
Okay.
Let's see. You have it there?
Yep.
Okay. This is the first spreadsheet, and you will notice that these are the patients that have been diagnosed with ASD. And you'll notice the column called Special Diet. And every single one of these things, of these subjects, are on some sort of a special diet. GCFF, GFCF, I always want to say it wrong. GFCF is gluten-free, casein-free. And you'll notice some of them are, soy-free, corn-free, dye-free, sugar-free. I forget what SCD stands for.
Yeah, what is SCD? I really wanted to know what that was.
I don't remember. Isn't that funny? Somebody could quickly look it up. I don't know. And you'll also notice they're also taking a whole bunch of nutrition.
Carbohydrate diet.
Pardon?
Specific carbohydrate diet.
It's
yes.
grain-free, sugar-free, starch-free, and unprocessed. So basically raw.
Right. And you'll notice they're also taking, most of them, excuse me, are taking some kind of nutritional supplement as well, many of them. Okay, now let's look at the other spreadsheet. Now remember, this is the autism group. Now let's look at the other spreadsheet, the control group. That should be the next spreadsheet, Cliff.
Oh, why is that not showing?
Try sheet 1. No, okay. No. It's a different spreadsheet altogether.
Oh, right. Okay, give me a second.
Okay. It should be the very next spreadsheet.
Yeah. Just got it now.
Okay. Yeah, demographic and medical history for non-ASD controls.
Yeah, is that the one?
No. Oh, wait. I clicked on the wrong thing. Yeah, that's it. Okay, now you will notice that I'm sure you will notice the column for diet and the column for nutritional supplements because they aren't there. Mm-hmm. So in other words, we have no idea if any of the control group were on special diets or if they were taking any nutritional supplements. Perhaps they weren't, but it would be a good idea to kind of mention that. That's missing altogether. And so you've got two different complete data sets, and you can't compare properly with two completely different data sets.
Medications as well going on.
Well, they do say multivitamins under medication, which, yeah, okay. The thing is, when you've got people on special diets, gee, do you think that could affect your gut? Mm. Do you think that could affect your digestion? Mm-hmm. But one of the things I wanted to look at here is I'm gonna screen share for a minute, and I'm just gonna briefly show a couple of where's this stupid thing? Okay, here we go. A couple of studies that I looked at. I looked at a lot of studies. Okay, I don't know if you can see this one right now. And darn it if I can't see the one I'm trying to look at. Okay, here we go. It doesn't seem to be working properly. Blast. Well, anyway, forget that. Let me just stop screen sharing. Anyway, I'll just read it. So I looked at a number of studies because, as Fiona knows, I'm pretty good at looking stuff up. I look at a lot of research papers, and this is one called Gastrointestinal Abnormalities in Children with Autistic Disorder by Horvath et al. And the thing is, 36 children aged 5.7 plus or minus 2 years. And another one is Abnormal Intestinal Permeability in Children with Autism. And we did a test on 21 autistic children. And it's not that important that I show them or not show them. The point is, one of the common things I found in looking at all these papers is the small sample sizes that they used. And I could not actually read most of the papers because they're behind a paywall. I do have subscriptions to both Science and Nature, so I can read any papers in Science and Nature. But there was a dearth of papers with having anything to do with autism and gastrointestinal disorder in Science or Nature. So my conclusion is they just didn't bother to submit any to them. I wonder why. The closest I got was the thing I've already mentioned, where desperate parents spark search for new treatment in Science, where they were talking about the story of Victoria Beck and the taking the drug secretin, which, as it turned out, didn't do any good. And there are a list of papers in Nature that relate to autism and gastrointestinal, but nothing, no smoking gun here. But what I wanted to show you is, Cliff, that one I showed, the one called
oh yeah, the page after.
Yeah, that was called Diet and Autism. Just go ahead and show that, but I'm not going to talk about that right now. So I had to basically go to a lot of people analyzing various things rather than the papers themselves. and to try to see what's going on, one of the things I noticed is that most of the proponents of diet and autism were medical doctors. Most of those objecting to the connection between diet and autism were researchers. And the reason for that is simply as when I looked at the this one article by a medical doctor, he pointed out that what's happening is he has patients come into his, come into him and talk about, they're saying that they're having this. So, and it's like Brian Udell, MD, Director of Child Development Center of America, and he was talking about it. And he said the most common GI symptoms include chronic diarrhea, abdominal distension, discomfort and bloating, et cetera, et cetera, et cetera, things we've already talked about. And, he pointed out that he sees a lot of the people. He says about 25% of my patients find relief and improvement with the GFCF diet. But remember, this is a apocryphal report. This has not been tested. When you look at the tests, they come up either non-specific with no specific answer, or they come up with there's no connection between diet and autism. However, one thing that has been pointed out by several people, including some on WebMD, is that even though there may be no connection between diet and autism, or autism and gastrointestinal disorders, no direct link that they can find, that parents who feel that their children are getting better should be supported. And this seems to be largely patient-driven. Now let's go to this article in Psychology Today. I'll put the link in the outside chat. I think it's a very good article because it starts by explaining that researchers have long disagreed about whether gastrointestinal problems may underlie some symptoms of autism spectrum disorder. It's not stopped some researchers and celebrities from promoting theories about a special autism diet. Now, this comes from the Harvard Mental Health Letter in April 2010. And he goes, wow, that's harsh. And he has several posts on this, and I would encourage people to read them. But it says Harvard Mental Health Letter is blunt. There is not enough evidence to support special autism diets that eliminate casein or gluten. And he points out the previous studies were not properly done or too small or didn't even use standard measures to diagnose autism in the first place. Does that sound familiar?
Yeah.
Isn't that, what we're looking at? He said that there's only one double-blind crossover trial of casein and gluten-free versus regular diets. And he says, not sure how you really double-blind that, but okay. Of 15 children with ASDS, and after 12 weeks, the diet seemed to have no effect. Some of the parents felt that the intervention trial was more successful than the independent reviewers. And he goes, so we get the picture, Harvard Mental Health Letter. And he talks about what autism is and how difficult this is. Because first of all, what is autism? It's a wide spectrum of disorders that have a wide array of symptoms. And you have to back up and figure out what is autism before you can even decide how to, classify whether they have gastrointestinal problems or not. And he goes on to talk about that, he says, to be fair, the lesson best gluten-free, casein-free diet trial was published in April of 2010 too, and the leaky gut study I'm looking at came out in late 2010. And he says, wait a minute, back up. How does one measure a leaky gut? Well, to test the gut integrity, one can drink a solution of metabolically inert sugars, and so on and so forth. And a person with leaky gut, those sugars will end up in the urine. And he says, of course, celiac disease is also associated with inflamed leaky guts. So the researchers in the study took 90 kids with autism spectrum disorder and 146 of their first-degree relatives, and also 64 children and 146 adult controls. Everyone was given an IPT test. Some were checked for FC, and all the kids with autism were screened for celiac. And so on and so forth. Said, sounds interesting. What are the results? Adult controls 4.8%, child controls 0%, ASD patients 36.7%, relatives 21.2%. Now notice this: the ASD patients had 36.7, but the relatives 21.2. Now I'm not going to go further into this. I'm going to suggest these papers to other people, and he says, well, that's a mixed bag. There seems to be a subgroup of kids with autism and their close relatives who have leaky guts, and that's something that I have seen in other papers, that there seems to be a some sort of possible genetic correlation between some of the intestinal problems in the patients with autism. I didn't see any research that said, well, let's take these kids with autism who have gastrointestinal disorders and study their relatives, and let's take these kids with autism who have no gastrointestinal disorders and study their relatives. So that a lot of research hasn't been done. So what's the bottom line to all of this? Is there any evidence? There is zero evidence that diets in any way whatsoever cause autism. Mm-hmm. There does seem to be evidence that a number of people who have been diagnosed with autism spectrum disorder do have some sort of gastrointestinal disorders. However, in a lot of parents who are reading all these autism sites, and I've read a bunch of autism sites, are reading about how these various diets help their children. So one of the questions I have is, are the parents giving the children these diets without any medical knowledge? And sometimes I know parents do this stuff without bothering to even tell their physicians. The reason that some of the kids are having gastrointestinal disorders is because their parents are screwing with their diets because they're autistic? So let's see, my kid's autistic, so I'm going to try a bunch of diets, and oh dear, now my child has gastrointestinal disorder.
Yeah.
The other thing is that
go ahead.
Autistic children, some of them quite faddy in what they choose to eat and not eat.
Yes. That's absolutely true. They are, and however, of course, the hype, the woo, the craziness continues. There is a group, and I'm going to give you the name of the group, called the Institute for Responsible Technology. If you ever hear of this group, be wary. It's not at all responsible. They think that GMOs promote autism.
Yeah. Oh.
So we now have this added to the mix.
Mm-hmm. Well, I've got this beautiful thing to add, and it's where I came across this article. It was shared by the atheist chef who asked for some input on it. And he had found it being referenced in an anti-vaccine article. Mm-hmm. Now, nothing that we've actually talked about with regard to this paper even mentions vaccines. Not one bit. And yet it was being referenced in an anti-vaccine article. And this is where I see the danger lying. It has nothing, the paper has nothing to do with vaccination. Nothing. And yet it's being used that way. And that's one of the reasons why I thought it was important to dissect this paper, to have a look at it. Now, the limitations on the study that we've actually pointed out this evening are actually acknowledged by the authors of it. They point out some of the problems with the sampling, with the age range, the split in sexes, etc. They point out these problems. But if you don't read the whole paper, you don't see that. You see the abstract, and if you remember, the abstract made the claim that they had demonstrated it. Does anybody remember way back then?
Yeah.
They actually stated that in the abstract. So if you don't read it any further than the abstract, you wouldn't even come across the author's own admission of the limitations of this study. So we're not sitting here tearing the paper apart. We're highlighting something that the authors have already pointed out. We've come up with some things that they also didn't really point out. And I think we've been a bit more vocal about the way in which some of these factors have impinged on the data that they collected. I myself would be saying, dinna publish. Just dinna. Gonna no do that. Just gonna no. And I mean, it's good that they have been willing to list those limitations, but it's not good that should you read no further than the abstract, you will not see that. Mm. That reading a paper isn't sit down, read the words, and that's you, you've got it. You need to understand what they've done, how they've done it, why they've done it. And I think that's what I want to be the take-home message from this hangout. That I'm not out to absolutely rubbish the paper. I'm not out to rubbish the authors of the study. I'm trying to point out the importance of reading the whole paper and understanding what it is that you are reading. If you don't understand those intricacies, then you are not actually going to get from the paper what it says. You'll get from it what you think it says. Now, I've come across this phenomenon myself when I started reading up and stuff when I was doing my PhD. There were some papers I read at the beginning of my PhD and I hadn't had to revisit them until I was coming to the end of my second year. And I read them again. And being much further into the subject by then, those papers suddenly weren't saying what I thought they said in the first place. They were saying something different. It was much more significant. And it was only because I knew more about it by then. I think that's something that we say to people, go and read the literature, see what it says. The literature doesn't support you or whatever. And we do forget this, that some people aren't in the position where they can go and read the primary literature and understand what it's saying. I mean, we've got somebody in here, we've got concordance in here, explaining all about the microarray problems and the qPCR problems. And we get Muggins here as well. And we get Jade talking about the dietary problems. We had Hogtie talking about some of the problems with procedures, etc. It's taken more than one specialism to explain it all. And yet we ask lay people to go and read the primary literature. Now, we've got things like New Scientist, Scientific American, that translate the primary literature into more digestible formats. But even then, I think people can end up coming away with the wrong message. Sometimes New Scientists can do that themselves. I'm not trying to be elitist here, but I think when you want to know what the scientists are saying, you really need to ask the scientists what they're saying. Because nobody can tell you what the scientists meant to say.
You're absolutely going to be accused of being an elitist for that. But I don't know. I find it strange that people. I mean, it's nice when scientific information is accessible, and science is one of the few fields where it's actually one of the pillars of practice. Part of the reason you become a scientist is for communication of information to the public. But you know, to watch some people argue with what is being reported as scientific findings, I've kind of drawn the analogy before of, like, you know, somebody taking his car to a mechanic saying, "Oh, I think there's a problem with it. I think the valve that you know, where the, you know, when the coolant and the oil mixes and then it goes to the separator, you know, I think there's something wrong at that stage." And then the mechanic is like, "No, like the oil and coolant don't actually mix. They're in separate channels." And I said, "No, they do mix." No, I'm a mechanic. I've been a professional mechanic for however many years, and that's just not how an engine works. And they go, "Okay, well then I'm not going to listen to you because you don't agree with my opinion about how an engine works. I'm going to go talk to somebody else." And people are so quick to saying, "Oh, my cousin's a mechanic, and this is what he says," and they'll accept that expertise. And yet, when it comes to interpreting scientific finding, there's this view that, oh, but if only we would just go, read the abstract ourselves, and it's. I mean, even as you're tallying people's contribution to the conversation, I mean, I like to think that the most important thing I said in this conversation was, gee, had this paper been sent to me as a referee, I would have said no, thank you, because I just don't know, like PCR, like what is this stuff, and there seems to be this liberty in like, well, I can skim the abstract and understand what was going on here. And then in terms of the accessibility of the primary sources, the publications themselves, there's a real irony with what the internet has done, in that it seems that the articles that are more accessible to the general public, remember the general public, as Sam Harris has pointed out recently, don't want to pay $2. They don't want to have a deal with a paywall where you're going to pay $2 for something. These articles are often $35, $40, $45, to get your own PDF copy of it. The paywalls are pretty steep for people, and the irony is that the ones that are the more accessible to people, that is free, tend not to be the good journals, where you can do the proper research, go and pay your $40 for this article, and then see that, while of the 40 references, there are 6 that are really critical to understanding. So I'm going to go seek those out, and that one's $25, and that one's $32, and a person trying to really educate themselves about the content of a single research article would be looking at a multi-hundreds of dollars investment just to have the PDFs available on their own hard drive, so that they can refer to them. Now, conceivably, we could set up a little research group and say, well, we're going to go read these things, and we'll only have to pay this fee once, and we'll all share these articles amongst ourselves, and we'll put them up on our, we'll put them in drop boxes, and we'll link them to these Hangouts so that you can go read them yourself. Well, guess what? We're not allowed to do that. You're violating copyright when you do that. The system itself makes it so that these things are not accessible to the public. So it's just full of ironies, and I'm sort of pointing out problems without really suggesting what the solution would be. I mean, what am I suggesting? We just get rid of paywalls entirely? Well, then, I don't know.
I did want to add something to what Hogtie is saying, because I think it's important. I live in a little county in California that's the smallest county in California, not counting San Francisco, which is a county and a city at the same time. And we don't have a huge economic base here. We're based on tourism, but it's not like Disneyland tourism, and mostly agriculture. So we don't have a lot of money. And yet our local library, if you go down to the library here, the county library, it has access to all these journals. I can go down to the library and electronically access just about everything out there. And this is something most people don't know. If my little county has access to these journals, I'd be willing to bet most places in the United States have access to these journals, because this is one of the wonderful things about having everything digital now. So if people want to know, I suggest you check out your library, and the librarians are trained that most of them have master's degrees in library science, although they also have volunteers working there. But, so if you don't know how to access something, ask your librarian. You can take a link, take the name of a paper or whatever, and go, or a list of papers, go down to your library and ask your librarian to help you find them, and see if they don't have those journals, and if they don't have the journals, where you can find them. Many people live near universities and or state colleges, and they also often will let you. I can't speak for every state, but I could go to the University of California at Santa Cruz, not far away, and I can look up this stuff. In some cases, you need to get your own library card, and even some big cities. I have a library card to the library, the San Francisco City Library, or it's actually a county. It's a county library, even though I don't live in San Francisco, because they just let everybody have a library card. So there are, you can, if you want to, you can probably find these papers free, but you have to put a little effort. I have a list of papers that I keep lists, and so periodically I'll go to the library to look up papers that I can't access from home, and there are some things that are in like the stacks, and I have lists of things that I can go to UC Santa Cruz and find out. So just a word to the wise out there, you probably can access the stuff.
Well, it gets actually better. If you go to a public library, if the librarian, you know, can train like.
Prudent, you've got a dreadful static.
Okay. Is this better?
No.
No.
Okay, resetting.
Yeah. When Prudent comes back in, you can get the floor.
I'd like to make a quick statement if I could before
Yeah,
he comes back. There's no reason why there wouldn't be GI involvement in autism. It's a perfectly valid hypothesis to have in the absence of other evidence. It's just odd how it is being used, or how the cross-link got made. The scientific data says one thing, and yet the popular media and the big autism conferences for parents, focused on parents, seem to report the results in a completely different way. That's the disconnect I have yet to really understand. Is how can you take a study that shows that there may be leakiness in the gut and transfer that to causality in autism? Again, I think it's not a terrible hypothesis that there might be a GI component to autism. We still don't know. There's so much we don't know about what is causative that I'm glad to see research into it. I don't think this is honestly come by. I don't think the intention here was to actually advance the knowledge. It seems to have just created more questions. And I think in some ways the guardians of the literature, the peer reviewers, need to be thinking about that kind of thing. Results that are tail-chasing exercises that make no real conclusions that can be well supported, they don't help anyone. I would much rather have had negative outcomes from this. We could not determine whether or not gut inflammation was involved. That would have been, I think, much more useful. The way they've sort of tortured the experiment to come up with some data, I don't feel as good about.
Yeah, and sorry, Prudent, I know you were next there, but just on the heels of this observation that it's not a ridiculous hypothesis, because even in cases where you don't have the so-called the general term that the leaky colon, where the products can actually get into the bloodstream because of breaches to the protective membrane in the colon. We're getting increasing information about the type of gut flora you have. So we all know we have these these we call the good bacteria that live in our intestines. Their waste products are small enough that they can actually go pass through, be absorbed with nutrients, go through the bloodstream, and even cross over the blood-brain barrier. Now, that's nothing to be alarmed about, because remember that the bacteria that you bear in your systems now is something that we've got a long evolutionary history with these bacteria. So if there was something really, really awful about having these bacteria around, I mean, it should have been dealt with by the evolutionary process by now. But that's not to say that all things never change. It might be the case that there are some cuckoo mutations, or some particular strain of bacteria that's problematic, and these are all really interesting hypotheses to entertain. But yeah, any research study that's using 8 or 12 kids in a control group and making claims about oh well, there's this, and therefore this is shown to have an effect on some neurological condition, is just as Concordance had said, it just doesn't seem honestly come by. I'm sorry, Prudent, I didn't mean to step on your point. I was just trying to catch Concordance on his point.
Oh yeah, I was just urging it to a point that Jade had made. You were saying that, yeah, like at the public library, if the librarian can train, because they're trained in trying to understand, or they're trained to teach people like how to navigate through a library website, so you can actually look up these papers. There's even more to that. If you understand like how to use the library database, you can actually access all of that from the comfort of your own home. You don't always have to actually go to the library itself.
Yeah, Prudent is correct about that. Depends on the library system, but you're right. I forgot about that. There's something I wanted to add. Something I just wanted to add to what Hogtie said. It's kind of interesting. It's very different but similar, because you're talking about the gut bacteria and how, yeah, it could possibly, be associated with autism. One of the things that they found out with some research now is how obesity may be related to the gut bacteria. They do that surgery, and I can't remember what it's called. I always get it wrong. The surgery where they reduce the bariatric surgery, something like that. Where they reduce the stomach or whatever. Well, the recent research shows that it may not be the surgery, it may be the change in your gut flora that causes people to lose weight and keep it off. Mm-hmm. Yeah. Which is just very interesting. So yeah, what's going on in our gut can affect a lot of things. But the point I wanted to make was originally was about the number of children who have autism and kind of how this reflects to this. Parents and a lot of people who don't understand what's going on are very alarmed with the huge increase in the number of children diagnosed with autism. However, research shows that all that's really happened is that one group of kids went from one diagnosis to another. It's not, and sorry I don't have that paper in front of me right now, but there is research to verify this. Basically what happened was you had a bunch of kids that had a particular diagnosis, and you don't see those diagnoses anymore. That doesn't exist. However, that group of kids is now diagnosed as autistic. So it's not necessarily that more and more kids are autistic, although that is possible, not excluding that. But we can see from the data that suddenly this one classification of children disappeared, and suddenly there's this huge increase in kids diagnosed with autism spectrum disorder. And one of the risks this runs, of course, is, as this paper I linked said, which what's an article, it's not a paper, it's just a very good one, and I think it has in it everything I researched, is that what is autism? Are we spreading this diagnosis too wide? That's something that people have been thinking about too. So you have to kind of narrow down the diagnosis before you can even begin to look at how seeing things like gut bacteria, things like food allergies, even relate to it.
Yeah. It is very interesting that the way autism diagnoses have changed. People are now coming under the umbrella term that would never have come under it before. As to a dietary component or a gut component, I've got nothing inherently against that. There isn't any evidence one way or the other, but I'm not opposed to it. I'll go where the evidence leads. And in fact, I've got my own little pet hypothesis, because of the regression factor that happened in autism, that there is something happening in these children that's environmental somehow. Whether that's due to a leaky gut that allows through a substance that just so happens to hit a certain part of the brain when there's a crucial development process going on, I honestly don't know.
Maybe it's just too much processed food. Parents.
That's the problem with processed food, because that's a really big group. It's going to have to be something more specific than that.
I mean, maybe it's a combination of a certain set of genes being turned on or off or whatever, and processed food. A combination factor.
I was going to say it cannot possibly be a single factor thing.
No, it can't.
Right? Like drinking too much windshield washer fluid is not good for your brain, right? And anybody who give too much windshield washer fluid to, you're gonna see the effects of it. So I mean, there are lots of examples of biochemical changes that happen in the body that makes a person susceptible to things that are not problematic for other people. I mean, we're talking about simple things like the lactose intolerance is such a simple thing where, up to a couple of years old, you can't really tell the difference from one person to the next, and then some people start having problems and they worsen and sometimes some pretty painful situations by their teenage years.
Yeah.
And that's two different. You say, well, that's the milk that causes it. No, it's not. That's the milk that triggers it in people that are susceptible to it. Mm-hmm. And it's the same thing with the processed food. You can say, well, it's the processed food or the power lines or point all this, but we're all living with processed food and power lines, and not that I'm saying anyone's actually suggested that power. I know people suggested that power lines have been the cause of cancer. Yeah. But we're all in this same environment to varying degrees, and a person could easily set up a fairly elaborate multivariate analysis of, well, this person lives this close to these power lines, this person eats this much processed food, this person does this much exercise, and then you could say, well, look, these are the people who get whatever condition of interest that we're talking about, and it turns out those kind of studies are never successful because there's got to be something that there are multiple factors to it. And wouldn't it be nice if it turns out that, that especially these more, severe and difficult forms of autism are a result of, as Fiona was just, pondering, like that there's some metabolite or byproduct of the processing of some particular foods that are have critical impact in some select group of people because of some combination of genes. Man, if we knew that, we could test people for that combination of genes or presence of enzymes or whatever the problem is, and then we would know. We would know when babies were born. We could tell the parents, hey, by the way, don't ever feed chickpeas to this kid, But, yeah. But that's not what we're facing. What we're facing is a media that is awash with horror stories about, for example, you just have to, you can compose these headlines with refrigerator poetry magnets. GMOs cause autism. Like just pick any two scare topics and put them together and say this causes that. Yeah. And people are going to latch onto it. We're sitting here giving people lessons on how, well, you can go to your library and you can get an account, and then sometimes you can access the stuff while you're at home and stuff. Well, the uphill battle here is the people that don't care how you might go about learning about how scientists communicate with each other, and will quite happily communicate with you. There's nothing elitist about the scientists, because the scientists will quite happily communicate with people. They sort of want to communicate in the way they communicate, but they're quite happy. Oh, you want to read our articles? Great. Which they didn't charge for it. But so that's the publishers that do that.
Yeah, I think the trick is to try and get the scientists to actually shut up once they've started.
I'm sure if I should.
Yeah, and what Hogtie said is very accurate. There are so many scientists, even famous scientists, if you email them, they will email you back, quite surprisingly. You find them at their university, you email them a complicated question. I have a friend who not long ago developed a really complicated question, sent it to a very famous scientist, and the scientist was thrilled with the question. And, my friend was like, gee, I wonder if I'll ever get an answer back, and they've had a conversation now. But I also wanted to mention something that I just thought of, that there's been quite a bit of research on, and that is the fact that we are living in too sterile an environment in many cases. That children who are exposed to various pathogens when they're young grow up healthier and even have have less cases of asthma, have less cases of some intestinal disorders. We were designed, quote unquote, excuse me, to live in the wilds, to live on a farm, to get our hands dirty, for children to be exposed to all those things so we could develop nice, fat, healthy immune systems. Children are being raised too sterile today. There are even movements now to get kids to a farm, to get them to handle chickens and things like that, and pet a goat or a cow, or to get out into nature and be exposed to these pathogens, because we're living in such a sterile environment that children are growing up, with
defenses.
Okay, sure, but okay, sure, but I want to be really clear, and I don't think Jane, this is what you're saying at all. I hate the way that observation about being around farm animals and getting outdoors and going for a walk and falling off your bike and scraping your knee and not immediately rushing and spraying some solvent on it to kill all the germs. Like, just be a kid, I love that. I love endorsing that. Not to be mixed up with, hey, take your kids to a measles party.
Yeah, definitely. Yeah.
What's said here is that we have, throughout our entire evolutionary history, or even if you're not into evolution, just the fact, as long as humans have been on the planet, call it 6,000 years, for as long as we've been on the planet, we have interacted with these microbial organisms a lot. And it's just part of how we are. And you need your immune system, you need to use it or lose it. And not that you lose it, but it certainly weakens when it's not being challenged. But it needs to be challenged by things that are mundane and routine, endemic to your environment. This idea that, well, we should give somebody measles because it helps improve their immune system is just so ill thought out. Because one of the things that measles does, like HIV, is it actually attacks the cells of the immune system itself and leaves it weakened for months. It's an incredibly dangerous thing to do to your kids. And these people who say things like, oh, you got to exercise your immune system. Yeah, exercise your immune system by going and petting a chicken or falling off your bike and not putting a Band-Aid on the scrape. I mean, saying that you're going to exercise your immune system by being exposed to measles is like saying, well, I want to exercise my biceps, but I don't want to do arm curls with little girly weights, so I'm just going to stand here and you can drop a piano on me from the 4th floor. Like, that's
I think to me a good way of putting it is, you want your kids to play in the dirt. It's good for your kids to play in the dirt, but please give them a tetanus shot first.
Yeah.
I was just going to say, when you said dirt, I was going to say, don't, Jade, tetanus. And you're already on it.
There was
that.
Tetanus
you were waiting. Yes.
The tetanus thing is pretty serious, too. Again, not to necessarily get into that, but it's got its own toll. Been a bad thing for our species, I will say.
So there's minors, there's all kinds of stuff in the dirt that your kids are going to be exposed to, and they're going to help build up their immune system. They do not need to be exposed to the tetanus. So it's not, it's like you said, it's not all or, use your sense, use common sense. Pet a chicken, but make sure that you're going to a farm that practices, oh God, what's it called? Poultry. Anyway, it's PP something or other. But there are farms that are very careful about how their poultry are handled. I have friends who belong to these groups, and just as get your kid out to play in the dirt. Let them have a great old time. Make sure they have the tetanus shot first.
Yeah. I mean, I crawl about my hands and knees picking up horse poo with my fingers, just because I'm so anal about how clean the stables are. I wish my horses were as anally retentive as I am. I was going to say something that would be really interesting, actually. I mean, you can't do it, but it would be very interesting to get blood samples from hepatic portal veins and see if there was anything distinctive moving from the gut to the liver in children with autism.
That would be, but until we have like the Star Trek transporter, we can transport the samples out.
So what kind of device are you thinking of hooking up to these children?
I know.
I mean, but I think that would maybe answer a question when we're looking at this leaky gut syndrome, because remember, all of the nutrients go via the hepatic portal vein to the liver. So if anything is being absorbed from the gut that shouldn't be, it should be there. It's just, how do you go about getting a blood sample from a hepatic portal vein?
Right. It's also just like, how do we run some of these tests when you can't have a control group, because nobody's going to experiment on children. Exactly. There are some things that just... we have to use animals. We have to go around the corner and use statistical data, and sometimes there are so many variables trying to get the exactly what is causing what out of all that statistical data. On top of which, if you're going to try to find the gene for everything, this is one thing that drives me crazy, especially when people say, "Well, show me a gene for homosexuality," and my first response is, "You don't understand genetics, because there isn't a gene for anything." One of my best examples I use for this is that, for example, if someone were to ask me, what is the gene for the color of a chicken's egg? My first response would be, well, that's a malformed question. Because there isn't a color of a chicken egg. It's two different layers of color, one on the other, and the result is what we see. What we're seeing is not what's actually there. You know, we're seeing everything from greens through even blues and golds and browns and dark browns and some kind of reddish that are really pretty. There's some chickens that have gorgeous eggs, into the whites and cream colors. So we're seeing this spectrum, but that's what we see. That's not the actual color of the egg or colors of the egg, on top of which there are different genes that are involved. And I don't remember that part. I've got to look up, or I ask a friend of mine who knows better. You know, I don't remember. For all I know, there's one gene for each color of each layer of the egg, or maybe there's a set of genes for each layer of the egg, or one gene for one layer and a set of genes for the other, because genes interact, and there isn't a gene for most things. In some cases there is, like red hair. That it's one change in a base, one change. It's a C to a G or a G to a C, I forget which, that causes red hair. But that's not always the way it is. And you have genes that do more than one thing too. So when somebody asks where is the gene for something, or where are the genes for something, that's a far more complicated question than they even understand.
Yeah, that is very true. Right, guys, I'm going to wrap this up.
Okay.
But I think we've done what we needed to do with the paper and shown why it's you can't just go by the abstract. And I just want to go back to one thing, and that was right in the abstract, given everything that we said about this paper. And I just want to read this out again. Taken together, these results demonstrate that ASGI children have a gastrointestinal mucosal molecular profile that overlaps significantly with known inflammatory bowel disease, yet has distinctive features that further supports the presence of an ASD-associated IBD variant. And I think we've shown that you can't draw that conclusion from the paper itself, given the number of flaws in the study. But that is straight from the abstract. Reading only the abstract, you would have accepted what that said and not realised the problems of the study. So I think the take-home message is always read the whole paper. If you don't understand the terminology, look it up or ask someone, and be more sceptical. And with that, I am going to end the broadcast. So say ta-ta, everybody.
Ta-ta.
See you, folks.
Ta-ta.
Ta-ta. Goodbye.